Hi everyone, welcome to topic 1 of our genetics unit. In this topic, we're going to be talking about DNA and its structure. So what is DNA composed of? DNA is composed of nucleotides, repeating nucleotides, okay? So a nucleotide is a structure with 3 parts. It's nucleic acid. Its 3 parts would be our nitrogenous base, our pentose sugar, and our phosphate group. In DNA, our pentose sugar is called deoxyribose, and we have 4 options for nitrogenous bases in DNA. So in DNA, the nitrogenous bases could be adenine, cytosine, guanine, or thymine. So DNA is made out of 2 strands, and these strands are complementary to each other, meaning that if one strand has A, the other strand will have its complement. And in this case, its complement is thymine. So adenine and thymine are paired together, okay? And guanine and cytosine pair together. Between adenine and thymine, we have 2 hydrogen bonds that form. And between guanine and cytosine, we have 3 hydrogen bonds that form. So you can see here's our nucleotide. We have our nitrogenous base, we have our sugar, and we have our phosphate group. And on the other side, we have a flipped version of our nucleotide. So this is what we mean by anti-parallel. DNA's anti-parallel is that the nucleotides are flipped of each other in the 2 strands of DNA. The side which the oxygen is pointing to in our sugar is called the 5' end of our DNA. So our 5' end is where our phosphate group is for our nucleotide and where the oxygen is pointing for our sugar. The direction that the oxygen is pointing away from is called our 3' end. Just to revise, why are they called 5' and 3'? Because if we count these carbon atoms, carbon 1, 2, 3, 4, 5, we can see that the phosphate is attached to the 5th carbon, so it's the 5' end. And if we start back here, 1, 2, 3, the phosphate is attached to the 3rd carbon, so this is the 3' end. Okay? So let's talk about hydrogen bonds. We said between adenine and thymine, there's 2 hydrogen bonds, and between guanine and cytosine, there's 3. What causes these hydrogen bonds? Well, let's imagine for a second we have 2 molecules of water. Okay? Oxygen really loves electrons. Okay, so the electrons hang out next to the oxygen more than the hydrogen. If we have more electrons near the oxygen, then it would be more negative. And if the electrons spend less time by the hydrogen, it's going to be a little bit positive. So these partial negatives, this little bit negative and this little bit positive of 2 different molecules, this attraction is called a hydrogen bond. Okay? So let's try this. In a water molecule, electrons spend more time around the oxygen atom and less time around the hydrogen atom. This causes the oxygen atom to be more negative and the hydrogen atom to be more positive. Okay? And then these attractions are called hydrogen bonds. So hydrogen bonds form between different molecules. That is comparative to a covalent and ionic bond, which are attractions between the same molecule. So this hydrogen is covalently bonded to the oxygen, and they're part of the same molecule, the molecule of water. This hydrogen bond is between 2 different molecules, a molecule of water and another molecule of water. So in this case here, in our DNA, our oxygen is slightly negative, and it's attracted to our partially positive hydrogen. And here we have the opposite. Our negative nitrogen is being attracted to our partially positive hydrogen. Okay, so let's take a look at our different nitrogenous bases that we have. We have cytosine, thymine, guanine, and adenine. We already discussed A and T pair together, C and G pair together. But cytosine and thymine only have 1 ring, so they are called pyrimidines. Guanine and adenine have two rings, so they're called purines. And I remember pure is long, right? So guanine and adenine are long. They're two rings, so they're more pure. And I don't know, I remember gap is purines. I don't know why. It's just how I do it. Maybe because CT doesn't make a word. All right. Okay, so this is our topic 1. Pyrimidines, A always pairs with T, so pyrimidines always pair with purines, vice versa. And the reason why that is, if we have two pyrimidines that pair together, in that spot, our DNA would be squished, they would be closer together, our strands of DNA. And if we had two purines bonded to one another, then our DNA would bulge out. Okay, so in order to keep it also a constant width, our DNA, the two strands, we have to have purines always pairing with pyrimidines. All right, thanks for